Welcome everybody to the March Corrigonan webinar series, and thank you for joining. All right, now I'm just going to advertise for April's webinar. Please note that it is not the first Friday of April. It is April 17th that we will have Claire Venny joining us and talking about epigenetic toolkits for fish. And I will drop the link in the chat to register for that.
Awesome. Thanks, Renee. And this is obviously not the first week in March either. So thanks everybody for being flexible. And so today we're really thrilled to welcome back to giving a webinar, Alex Gatch. Alex got his bachelor's in science from Hobart College up in upstate New York. Is that on Seneca Lake, Alex? Yeah, okay. And then he did a student services contract with the Lake Ontario Biological Station at USGS to get some experience, research experience, and then went to get his master's at Purdue University with Thomas Hook. From there, he went back to New York, where he worked with the U.S. Fish and Wildlife's Lower Great Lakes office. And then we stole him back at USGS, for which we're really happy that he's there. He's been there since 2023 as a research fishery biologist. And Alex, if you want to share your screen, he's going to talk to us today about a lot of work that he's been doing, some really cool experiments looking into a potential bottleneck or potential impediment to our bloater reintroduction in Lake Ontario, specifically transport stress. I think the talk is around between 35 and 40 minutes. So sit back, and Alex is kind enough to stay afterwards and have a discussion, answer some questions. So we look forward to that. Thank you, Alex.
All right. Thanks, Bo, for the invitation. Can you hear me all right?
Sound great.
All right. Thanks. Thanks, everybody, for joining me on a Friday afternoon. I'll be between 35 and 40 minutes, so I'll try not to steal your entire afternoon. But we have some exciting results to share with you in a project. We're trying to define what's killing the stock bloater that we've been reintroducing into Lake Ontario. So before I begin, I just want to thank the co-authors that are on this presentation and that have been with us for the extent of the study. We have folks at USGS, the partnership with U.S. Fish and Wildlife Service. Some of the bioanalytics I'm going to share later was run by a non-government bioanalytic lab. So thanks to Stress Bioanalytics. And then the funding from this project came from the EPA through the GLRI Corrigonan template. So thanks to all those co-authors and thanks to the funders as well. Okay, so we'll jump right into it, start talking about bloater in Lake Ontario. So we're going to be talking about Corrigonus hoyi today, and that's bloater. This is one of the deep water corrigonines in Lake Ontario. It would have traditionally been the historic forage for lake trout. And then it also had some ecological implications for nutrient transfer, energy transfer from deep water to some of the epilimnium as well through DVMs. And so this was an important prey species, but also for the ecosystem. All too well with most of our native species, at least in Lake Ontario, we had population declines that were pretty well documented starting in the 1900s and ultimately leading to extirpation by 1983. That was the last time we caught a wild bloater in our annual trawls. So with that in mind, Lake Ontario's management objective has been to both increase prey fish diversity, including bloater, but also to fill this empty deep water niche that was left by the extirpation of this species. So moving on to the reintroduction efforts, this was a partnership with Fish and Wildlife Service and others, starting in Lake Michigan in 2011. So Fish and Wildlife and others went out with the commercial fishers to be able to collect wild adults for gametes. So that started in 2011. And then since 2011, we've had a stocking program in Lake Ontario, both with our partners in Ontario and on the US side as well with Fish and Wildlife Service. So we've been stocking every year since 2012. We have stocked around about 2.5 million, and those have come from different life stages. The majority are age 1 fish that are released in the spring. But we've also had some egg releases, larval releases, and then a few adults as well. This data here down in the bottom right corner is some results from Brian Weidl's spring prey fish et al.'s spring prey fish report, and we can see that we've had 31 recaptures of these hatchery origin fish. So the blue points there show you where they've been collected up until 2025, and then the red points were our most recent collections. And kind of important to note here, last year was kind of a banger year for us. We caught 10 bloater, which was the most ever in our annual surveys. And some of those actually came from gill nets too. So not just bottom trawls, but gill nets. So with that little spark of success there, ultimately we've seen pretty low post-stocking survival because we've had so few recaptures. And this has been ongoing for the last 15 years. So now I'm going to go into just a little bit of a dive of what's been done to measure post-stocking survival. I'm going to focus on 2 studies from the near past that used acoustic telemetry. And just to orient folks that might not be familiar, though I'm sure most people are, acoustic telemetry is able to track fish movements with acoustic tagged fish that then swims around the lake and is picked up by acoustic receivers so that we can get positional estimates and timestamps of where those fish were. So in the late 2010s, Natalie Kleiner et al. Put together a nice study where they were measuring post-stocking survival using acoustic telemetry. And what they found was that on the first day of release, they had about 60% survival. So right off the bat, we're losing about 40% of the fish they were putting into the lake. They monitored those fish for a period of about 12 days before they left the acoustic array. And they saw that after those 12 days, we had about 42% survival of the tagged fish. They also went a step further and started to assign where that mortality was coming from. They didn't have predator tags in this specific study, but they estimated about 11% of the tagged bloater. The mortality was coming from predation, with the rest of that, so 47% of their tagged fish was from natural causes. In 2025, this was part of Lydia Pollock's master's thesis. So her and others went out and added on to this study. They added predation tags to the tagged bloater so we could see like, oh, well, now we actually know that this fish was preyed upon. So this figure here on the y-axis shows you the probability of survival of fish. And then on the x is the time. So how many days since they were stocked. The other nice addition of this study is they stocked at different depths. So each one of those colors is yellow is 5 meters stocking depth, blue is 50 meters, and green is 100 meters. And kind of the takeaway there, at least in their 2022 study, was that they had extremely low survival. So between 12 and 30% over the first 3 weeks at large. And that range there is just based on which depth the fish were released at. The other important part of that study was that they saw mortality with 35% of tagged fish that they had was from predation. And they even split that between avian predation and fish predation too. So I don't have those numbers up here, but just know that not all of that 35% is from fish. Some of it's from birds as well. And then maybe even more interestingly, they assigned 19% of those mortalities to compression barotrauma was their hypothesis. So as fish are released from the surface and swimming down to some of these depths as 50 or 100 meters, they hypothesized that they're experiencing compression barotrauma. So that's where they assign that mortality. So using this information, 2 of these nice studies from Lake Ontario, some of us put our brains together and says, well, how can we control for this? And this was really led by Brian Weidl, who's at Lake Ontario Biological Station for USGS. And looking through the literature, we thought that maybe using some more soft releases, so this is acclimating fish to the lake before releasing them, would help increase survival. And so in thinking about this, we developed large net pens that we were able to deploy off of some of our small vessels. Excuse me, the net pens were cylindrical. They're 2.5 meters in diameter and about 10 meters in depth. And that depth is important because we were trying to confine the fish to 1 atmosphere's worth of pressure, so we could try to mitigate any type of compression barotrauma. We built 3 of those net pens and deployed them in slightly different ways. The first 2 net pens, we had stocked 45,000 bloater into each of the net pens, so 2 net pens with 45,000 each. The densities, because the net pens were so large, were very small, so there's only about 10 grams per liter of fish densities. And then in our third net pen, we released only 30 acoustic tagged fish. So imagine 30 fish in this massive net pen. These net pens were in about 40 meters offshore from Oswego, New York, so they were an offshore release. One of the goals of this project was to assess survival after release. So it wasn't necessarily, how long or what was the survival initially. It was being able to collect these fish again in trawls down the line, so years later, and then also using the acoustic telemetry to assign mortality after release. But what this allowed us to do was to control for predation. So now there's no bird predation, there's no fish predation for these fish. And hopefully we were able to control for compression barotrauma the best that we could by confining those fish to the first atmosphere in the lake. We also loaded the 2 net pens with 45,000 fish a couple different ways. We used the research vessel CAHO, which is Lake Ontario Biological Station's large vessel, to load fish. So in this case, fish were touched twice. They were touched once to get into the hatchery truck, and then loaded into the CAHO, and then loaded into a net pen. So there's 2 transfer events. And then the second method, we used a barge, which we could drive our hatchery trucks directly onto the barge and then stock the fish into the net pens using just a single transport event. Okay, hopefully this video will work, but I'm gonna show you a video of what the net pens looked like 24 hours after stocking. So a little bit of crazy ROV driving, but the net pens are still on the surface, which is great. That's what we had planned for. But we're starting to see a bit of mortality within the net pen. There's still quite a few fish swimming in the net pen, which are seemingly fine, but you never actually know what's going on in there until you release the fish. Okay. So what did we find with the net pens? Depending on how the fish were loaded, there was a little bit of a difference in survival. And these are really just estimates because it's pretty hard to quantify how many out of 45,000 fish died in a net pen, especially because the point of this experiment was for survival, long-term survival. So we didn't want to count individual fish. These estimates are coming from taking pictures and then subsampling the pictures to then be able to quantify what we think was as far as mortality. So right around 50 to 75% survival after 5 days before the fish were released. And then importantly, we noted when we went out in successive days to ROV footage, the majority of that mortality was within the first 2 days of stocking the fish into the net pens. So long-term, Brian Weidel is still working up this data because we're still collecting bloater, but the plan is to be able to quantify survival after these fish were released and compare that to the traditionally released fish, which occurred at the same time in that year. So what about predation and barotrauma here? There was no predation within the net pens, and barotrauma was controlled the best that we could. But we still saw, pretty large mortality event within the net pens. Going to the second experiment here. So this is the third net pen that had just 30 acoustic tagged fish. Kyle Morton at U.S. Fish and Wildlife Service worked up this data for us, so thank you, Kyle. But what he found was 63% survival after 5 days. So this is while the fish were still in the net pen before they were released. The figure here shows you survival probability on the X, and the Y again is time after release. So we paired this with fish that were traditionally released. So there's also 30 acoustic tagged fish there that were not in the net pens. And regardless if you were in a net pen or not, you still experience pretty strong mortality in that first couple of days. And then when we looked at survival across the time period, so about 30 days post stocking, there was slightly higher survival in the net pen fish, but it was not statistically significant. So again, here we note that we were able to control for predation and barotrauma for the most part, but we still saw this steep decline in survival in the first couple of days post-release. So to just give you a conceptual diagram of where we think the majority of mortality is coming from, this is a simple timeline. So fish are loaded at the hatchery. This is time 0, so 0 hours. They haven't been transported yet. There's really no mortality from transferring from tanks to the tank truck. Those fish are then transported 4 hours. That's how long it takes for us to get from the Lamar National Fish Hatchery in Lamar, Pennsylvania, up to Lake Ontario. And again, there's really no mortality within the trucks themselves. They're checked every hour on their trip up to Lake Ontario, so we can control things like temperature and dissolved oxygen, and we really don't see mortality there. Once the fish are released, and about 24 or 48 hours after the release, we lose effectively 30 to 60% of those fish at this time. We don't really know why that's happening, but that's what we're going to talk about today. And then after about 2 weeks at large, we do see more predation, and that's been supported by some of the telemetry analysis that's been conducted. So we're really going to focus on this part today because it seems to be the most important. This is when we're losing the most fish in the stocking event. So that brings us to the question, is physiological stress from transport causing post-release mortality? We know that it's not within the truck while they're in it, but what happens after those fish are released, and was there some sort of stress that's adding on to what they're experiencing in the lake? Transport and handling stress is very well studied in other species, though it hasn't been studied, at least not published, for bloater. And the mechanism here is that these stressors can cause imbalance in homeostasis, and this affects things like the immune response of the fish, the growth, behavior, swimming efficiency, and importantly, exhaustion once those fish are released. Another important piece of information that we got thinking about transport and handling stress is that across the board, no matter which hatchery manager or technician you talk to, they notice that bloater are much more delicate compared to other species that they've raised in the hatcheries, and they can respond poorly to changes in their environments. This is kind of a clue that maybe something is happening here as far as stress that might be causing some of the mortality. Again, looking into the literature, there are certain levers that we can pull from hatcheries in transporting fish to mitigate stress and hopefully have an impact on survival. One of the really easy ones is to change the time at which you stock fish. So either during the light hours or in the dark. Others have found that by releasing fish in the dark, you can increase survival. Another easy change is to change the transport water temperatures. Others have found that colder water temperatures can reduce stress in fish during transport. And then the final one, which is again pretty simple to change, is how do you handle the fish? Are you using nets or using pumps? And others have found that keeping fish submerged rather than dewatering the fish can also reduce stress and increase survival. So with that in mind, we have 3 study objectives that I'm going to talk about today, and they're quite simple. The first was to determine if there even is a physiological stress response to transport and stocking. The second was to test those different factors. So stocking time of day, transport water temperatures, or handling a type and how they might affect the stress response. And then the third was maybe the most important was, does post-release survival change with water temperatures and handling? So did the fish actually die, or is it just a stress response? To do this, to be able to quantify stress, there's a couple of different ways that folks have done it in the past, but the one that's probably the best correlated to stress is measuring blood plasma cortisol concentrations. Cortisol, same as in humans, this is a hormone that's released in response to some stressful stimuli. And it aids you in your fight or flight. So basically what it does is release a bunch of glucose in your blood, gives you more energy, you're more focused, which is good over a short term, but chronic stresses caused by these upregulations in cortisol can have some downstream effects that are not necessarily beneficial. Looking in literature, there's also quite a bit of information to suggest that if you handle fish within 3 minutes, you're going to start spiking cortisol. So it was important for us to do this really quickly for each fish. So we took blood samples in less than 3 minutes once those fish were handled. We separated the plasma from blood samples and froze those samples in -80 freezers, and then sent all of our samples to Stress Bioanalytics, which is a bioanalytic laboratory in Oswego, New York, where they used ELISA assays to be able to give us cortisol concentrations from each sample. Okay, so to get into the nuts and bolts of what we did, I'm going to talk about 3 different experimental designs that took place from 2024 to 2025. And the first one was to answer the question, does stocking time of day affect bloater stress response? To do this, we measured blood plasma from age 1 bloater. This is the kind of the norm for what gets stocked into Lake Ontario. And we did it with the U.S. Fish and Wildlife during their annual stocking. So this would have been typical to what a bloater would experience in any given year. Those fish were raised at Lamar National Fish Hatchery in Pennsylvania and then transported up to Oswego, New York, where they were stocked into Lake Ontario. We had 2 factors here for our analysis. So time of day with 2 levels, daytime, nighttime. Hours of transport was our other factor, and then the interaction between those 2. For as far as blood draws go, we had sampled 10 fish for every hour of transport, and we sampled those from hours 0, which would have been before fish were even loaded onto the trucks. This is just fish in tank, and then every successive hour until the 4th when fish were released into Lake Ontario. So we had 100 total samples there. This is also important to note here that all of these samples were taken within a single 24-hour period. So we drove down to Lamar early in the morning, drove back up to Lake Ontario to stock the fish, and then within that same day drove back down, waited until it was dark, and then did this. Took us about 20 to 22 hours to conduct this experiment, but it all happened in the same day. So those environmental factors in the lake were the same as well. Our hauling density was the standard for Fish and Wildlife. This is about 29 grams per liter, and they use a 0.5% salt solution. And we had about 3,100 bloater in each of our hauling tanks. Our analysis is very simple. We use a 2-way ANOVA with 2 keys HSD, and we can get into those results here. So to orient you to this figure, it's going to be the same for each of our experiments. On the y-axis, we have cortisol concentration, that's in nanograms per milliliter. And then on the x-axis, we have time step. So 0, again, is before fish were ever moved, and then each hour from there. The colors here are yellow for AM, black for PM, and those points there are averages with 1 standard error around them for the error bars. So what we saw was that cortisol concentrations did change significantly over time steps, with that first time step having the most significant change from 0. So after the first hour, there was no significant difference, even though there's a slight increase in cortisol. It's really that first hour that spikes the stress response in these bloater. We also found that cortisol concentrations did not differ between time of day. So it didn't matter if you stocked the fish in the AM or the PM, they still had a stress response. And there was no interaction between time step and time of day. So the takeaways from that first experiment, bloater did have a measurable stress response to transport, and it really just spikes after the first hour of transport, and then it plateaus. And then the big one there was that transport during the day or night did not differ in the stress response. Moving on to our second experiment. So this also occurred in 2024. We did this at the Tunison lab. So here we're testing the effect of water temperature on bloater stress response, and we also quantified survival here as well. We use the exact same cohort of fish. So, genetically, these fish were siblings and cousins to the fish from the original Fish and Wildlife Service stocking experiment. We did a mock 4-hour transport. So we simulated the same amount of time that these fish would have been transported. And again, this is from the Tunison lab, which is in Portland, New York. We had two factors here: water temperature, which had a cold temperature, which was 5C, and then 10C, which is the ambient water temperature that these bloater are raised on at the Tunison lab their entire lives. We had time step again, and then the interaction between those two. The blood draws in this case, again, we had 10 fish per time step, but here our time steps changed. So instead of going every hour of transport, we went before transport, immediately after transport, which was 4 hours, then 24 and 48. And we did this because we saw the peak at 1 hour in the first experiment and then it kind of tailing off after 4 hours. We replicated this over 5 days. So there's 5 individual stocking events, and that came out to 400 total samples. We tried to match the hauling density the best we could here. So 27 grams per liter with a 0.5% salt solution. And our tanks here were much smaller. So we only had 80 bloater per tank, and this just allowed us to have more replication because we simply didn't have that many large tanks to have 3,100 fish. The other important thing to note here is that because fish at the Tunison lab are raised at 10 degrees Celsius for the extent of their life, we had to acclimate the fish in the 5 degrees Celsius treatment over a month before we could do any of the transporting, because this would have been the first time that these fish would experience that cold of water. We did something a little bit different for analysis here. We used linear mixed effects models with our day as the random effect. So that day is the individual stocking event out of the 5 days. And then we also quantified survival. So after the stocking event, we brought these fish back to the lab, released them back into the tank that they had originated in, and then quantified survival every day just by counting the number of dead fish. Here we use the Poisson generalized linear mixed effects model, and again, use the day as our random effect. So just looking at this, it looks very different than a Fish and Wildlife stocking event because our sample sizes were so low for each tank, so we could have more replicates. We used coolers instead of a hatchery truck. So these fish were treated kind of the best that you could imagine. They were gently netted into these coolers and then gently netted back out. They weren't, shot out through a hatchery truck using water pressure, which would have been the general way that fish are stocked into Lake Ontario. So just something to note there as we get into the discussion later on. Moving into the results, so this figure is set up the same way with cortisol on the Y and time step on the X. Just remember the time steps have changed now. They're at 4 hours, 24, and 48. And then the different colors are the different temperatures. So we found that cortisol concentrations change over time. This is similar to experiment 1. That highest spike was right at 4 hours. And then by 48 hours, those fish have come back down to a baseline of cortisol concentration. The important difference here was that we found an interaction between temperature and time step. So cortisol concentrations were the greatest for the 5 degrees treatment at the 4 hour time period, and then again at 48 hours. So we did see an effect of temperature here within its interaction with time step. Moving on to survival, this was very different than what we thought we were going to find based on the studies that we previously have conducted in Lake Ontario. On the y-axis here, you have percent survival. So this is out of the 80 fish in each of the tanks. And then on the x-axis, we have days after transport. And we saw a pretty flat line here. So water temperature did not have an effect on mortality. And if you look at the scale on the y-axis, a couple fish died per tank after the transport event. So it wasn't this very large mortality event, which we see in the wild. So the takeaways here, again, bloater had measurable stress response. We saw that it was returning back to baseline after 2 days post-stocking. We also saw that stocking induced strong acute cortisol response in fish at both temperatures, but recovery was slower and the early stress response was more pronounced at 5C compared to 10C, which is a little bit counterintuitive to what others have found, with colder water temperatures generally having lower stress. In this case, our colder temperature actually stressed the fish out more in that first time step. And we also found that post-stocking mortality did not differ between the temperatures, and in general, it was quite low. Okay, moving on to our final experiment. This was conducted last year in 2025. And here we were trying to test the effect of handling type and how that might affect the bloater stress response and survival. We conducted this again at the Tunison lab. So we had the same age class of fish, so age 1 bloater, and they were put through another mock 4-hour transport. One major difference here is that we used outdoor raceways as our tanks instead of those much smaller tanks that you saw in the last experiment, and this allowed us to have more fish in each of the stocking events. We had 2 factors here again. One was handling type, so traditional netting was one of the factor levels, and the other was using a fish pump, which does not dewater the fish in our case. So they were kept within water the entire time while they were transferred from the raceways to a hatchery truck. And then we had time step again and the interaction between the two. We increased sample size here to 20 fish per time step instead of 10, like we had in the last 2 experiments. We had those at the same time steps, 0, 4, 24, and 48. And we had replicated the stocking event over 3 days. So 3 separate stocking events with a total of 480 total samples. Our hauling density here was pretty similar to the last 2 experiments. It went up a little bit just because we increased the number of fish in each tank. So we had 1,200 bloater in a tank compared to the 80 in the previous experiment. Because fish at the Tunison lab are raised in circular tanks indoors, when we move these fish out to the raceways, we needed to acclimate them. So we acclimated over 2 months, but the water temperatures between the 2 areas the fish were raised in were the same. So they're really just acclimating to different densities of fish in tanks and a different structure. So concrete instead of fiberglass tanks. Our stats here were the same as the last experiment. We're using linear mixed effects models with Day's random effect. And then our survival analysis was the same, again, with the Poisson generalized linear mixed effects model. This gives you an idea of the 2 different handling types. We used an Aqualife BioStream pump, which has a 2.5-inch transfer hose. So those fish were suctioned from the raceways into a hatchery truck here. So we went back to a traditional 250-gallon hatchery tank that the fish were transferred into. And then we compared that to traditional netting, which is exactly what it sounds like, using crowder bars to crowd fish and then netting those resulting fish into the hatchery trucks. So those fish were dewatered where the fish pump bloater were not dewatered. Moving into the results, same structure here again with cortisol on the Y and time step on the X. The blue color is the pump and the yellow color is the net. We found that cortisol concentrations differed significantly over time, similar to the last 2 experiments. And then looking at this figure, I have to orient you here because it's a little bit hard to interpret the way we have it with time step at 0. We found that cortisol concentrations differed between the 2 treatments, but it's a little bit different than you may think. We're not sure what caused it, but at our first time step before fish were ever moved, we had 43% higher cortisol on average in our pumping treatments. So they weren't apples to apples from that first time step. And when you look at the percent increase between time step 0 and 4, there's actually almost twice of an increase in the netted compared to the pumping. So within this interaction, pump fish had lower cortisol increases at that 4-hour, and they also had faster recovery at the 48-hour. So that's kind of hard to think about when you look at this figure just because you see pumping cortisol so much higher, but it's really less of an increase just because of where they started at their baseline. Next, looking at survival, this was pretty similar to our second experiment. So percent survival is on the y-axis here, and X is days after transport, pretty flat line. We did not see a significant effect in mortality between those two treatments. And again, we're talking about a tank of 1,200 fish and only 1 or 2 fish experiencing mortality over a course of a week after transport. So quick takeaways here. Bloater again had a measurable stress response and it returned to baseline after 48 hours. This is the important one here, that bloater in the pumping treatment had higher initial cortisol. So again, it was about 43% on average higher just off of baseline compared to the netting treatment, but it was associated with a lower stress response in that first 4-hour transport, and then a more rapid recovery after 48 hours. And then also we didn't find any mortality differences between pumping or netting, and it was quite low as far as mortality goes after the stocking event. Okay, so that was a lot of information all at once. Let's discuss. Going back to our objectives, our first was to determine if there was a stress response at all from transport. And this across the board was yes, there is a stress response. It seems to peak at that 4 hours of transport and then returns to baseline after 48 hours. When we looked across each experiment, the kind of average max cortisol was around 50 nanograms per milliliter. So we're pretty confident that each experiment was measuring in the same way. And this kind of brings up the question, well, is there a lethal threshold? What don't we want to exceed before we start seeing increased mortality? And it's pretty hard to answer that question, especially when we look through the literature at other species where this has been conducted for decades. As far as chronic stress goes, this is usually what kills fish. So fish are generally resilient to an acute stress, a quick acute stress. They can recover from that. But if you add on chronic stress, we start to see 500 nanograms per milliliter in other species is where mortality can occur. Going on to our second objective, this was determine those different factors, if they had any effect on physiological stress. The first was time of day. That had no effect on stress in our experiments. The second was looking at water temperature. We found that colder water temperatures created a greater stress response, and it took bloater longer to recover, which was different from what others have found. And maybe something to explain this here is allostatic load, which is kind of like the wear and tear of stress in someone's life or in a fish's life, could have been added on from the acclimation period. So remember, at Tunisia, fish are raised at 10C all the time. That's just the temperature that they're raised at here. We had to acclimate fish to a 5 degrees Celsius tank. And so that may have lowered their threshold as far as stress goes. And they might have been more prone to reactivity and stress once they hit that cold water and started to be transported. And then finally, our fish pumps created a lower stress response within that first 4 hours and had a faster recovery after stocking. So this might have been associated with things like less scale loss, because nets, the abrasion from a net might create scale loss. They kept fish in water, and we had faster loading times for the fish pump. You could load fish in under a minute, where it took a couple minutes to net the fish. And so all of these might have compounded to create a lower stress level for these fish as they're transported. The last objective was to measure post-release survival. We found that neither hauling, water temperature, or handling type affected post-transport survival in a laboratory setting, with the caveat that fish in experiment 2 were handled very gently, so it wasn't necessarily the exact same practices that are used in Fish and Wildlife stockings. But in our third experiment, we tried to replicate everything exactly the way that the Fish and Wildlife Service does, and still saw very low mortality in any of our stocking events. So this brings up the point that mortality generally comes from compounding chronic stresses and not necessarily one acute example of stocking. So what we think might be happening in Lake Ontario is it's a cascading effect of stressors. So once these fish are initially stressed in the transport event, that doesn't seem to kill them. but cascading stressors like predators, different water temperatures, different water chemistry, things like this may be contributing to mortality, but we weren't able to measure those in these experiments. So to conclude, the question that we all wanted to answer is transport stress causing this low post-release survival? And as far as we can tell from our controlled laboratory experiments, not by itself. So we think that the conditions, as I just stated, in Lake Ontario may be compounding on the stress from transport. We found in our stress experiments that stress peaks at 4 hours and then comes back down to baseline at 48 hours. But is that really what's happening in the lake? Probably not. There's all these other added factors in the lake that may be pushing those fish over the ledge. And it's important that if we can take any different mechanisms to reduce stress in transport, that they may benefit survival down the line. We just weren't able to measure those in this experiment. So thinking next steps, if anyone wants to, I'll hand over the torch on stress experiments to you. You could repeat these using mock stocking into net pens, where we can then continue to measure cortisol after the fish actually acclimate to the lake. Or again, we could use acoustic telemetry to answer some of these questions. But the ultimate thing to test next would be to consider in-lake rearing. And this would just completely eliminate transport and handling stress. So this might be an avenue where we want to look at next. With that, I took up 40 minutes of your time, but I'm happy to stay on for any questions.
Fantastic. Thanks very much, Alex. Really comprehensive talk. You explained it really well. I enjoyed your graphics. Yeah, I mean, you've got me convinced that transport stress in and of itself is not sort of what we might thought it was after the net pen conditioning experiments that initially caused so much concern. So at this point, people can ask questions either through the chat. If you want to write them in, or ideally, you could just flip your camera on, raise your hand, and ask Alex in person. Maybe while we wait for the first questions to come in, kind of going back to that conditioning idea that you and Weidel and had through the literature. Do you think conditioning can be helpful, and is it worth sort of repeating those experiments to see if, I mean, that's taking away the predation in terms of a compounding stress effect, but maybe the change in water chemistry, you had that as a source, and maybe the changes in water temperature over that 48 hours is more dynamic, and maybe not like, do we know what the water temperatures were in that net pen to know if that could have been a compounding factor as you sort of described it?
Yeah, absolutely. We had put hobo loggers within the net so that we could track that. And, we're always trying to match water temperatures at the hatchery to the lake so that we're not shocking fish too much. But these fish are stocked in April and May. And, the first couple meters of water might be 10 degrees Celsius, but everything else is 4 degrees Celsius. So those fish had 10 meters of play in there, and the fish that swam down might have been in 4-degree water, and they might have been at 10 degree in the surface. So it's kind of hard to say. We also had storm events that pushed the net pens down. So they swing kind of back and forth throughout the water column. We have all that data. It's something that we can definitely look at. But to answer the first part of your question, once we get back some of the results that Brian's working on as far as the fish that have been recaptured since those net pens, if there's any differences in survival between those and the traditionally released fish, that might push us to the next step of, implementing this on a larger scale.
What did you say that unique mark would have been for the net pen fish for you to be able
to
test that?
I didn't even mention that. Yep. So they're double OTC marked compared to a single OTC mark. Yep.
Okay.
Yep.
Awesome. Ted.
Nice presentation, Alex. I was out on the first couple boats that we started collecting eggs long before we started putting hatchery folks on there like we should have. But just wondering, I know there's a lot of pros to stocking them in the waters where they should be, deep waters that are cool, that kind of thing. But given the concerns about barotrauma and that kind of thing, and I guess the temperatures you just mentioned that may be closer to the rearing temperatures, would it be advantageous to stock it near shore, do you think? Or do you think the predator load on the way to where they're going to be or where they're going to be heading is outstrips that benefit? And I don't know if this is your ball of wax or not, but just wondering in terms of, it was really interesting to see all the stress stuff and just trying to think of, the boat's the boat transport out the middle of the lake, I imagine maybe somewhat stressful, but just wondering if you think a shallow water stocking is more advantageous than a open water stocking, given kind of what we've seen in terms of mortality.
Yeah, it's a great question. And I think Steve Davis is on here too. So Steve, chime in when I'm wrong. But the majority of stocking right now is from shore. In the past, it had been from the Caho and also, barges when they're available. The only problem is that lake trout stocking and bloater stocking kind of occur at the same time. And so it was like one versus the other to use the barges. And we don't have the Baird on Lake Ontario, so we don't have a vessel that's set up to do it that way. So yeah, I don't know, Steve, if you want to add to that, but I think the traditional method right now is shore stocking, and some of those telemetry, and like Lydia's study showed that there was like the random barotrauma mortality was higher in the 100 meters compared to the near shore, but the near shore had higher predation. So it's like, which one do you want? Do you want the birds to eat all the fish, or do you want them to all sink to the bottom and die? So yeah, I think it's a trade-off.
Yeah, and you're right. We are doing the short stocking now, and actually we're stocking next week, and Allegheny's putting their bloaters in the mouth of the Niagara, and we're putting ours in the mouth of the Genesee. So the last 2 years we've been doing at the mouth in rivers. BC thought that would be a good plan to try. So that's what we're doing now.
All right, thanks, Steve.
Yeah, and Steve, is the idea there that they're sort of more or less maybe pushed away from the near shore and not like washed back on the beach? Yeah,
I believe so. Yeah, I think it takes the wave, the weather and the waves. As Alex saw on the one night stocking, the weather was calling for calm waves. We got up there and it was a little choppier than what they were calling for, and caused some problems there as well.
Yeah. All right. Question, Ted. Thank you, Steve. In the chat, Alex, I don't know if you can see it. There's one from Katie Anweiler wondering about the 0.5% salt solution. Maybe the rationale for that. And yeah. Can you read the comment? Okay.
Yep, I can see it. And Steve, you can stay on for this one too, because you can answer it better than I can. But that's just in transporting fish, that's a general practice for all species really that go into Lake Ontario as a way to kind of calm down fish, also activate them to produce more mucus. So Steve, you can talk more about that too. I'm not a hatchery specialist.
Yeah, I think you answered it pretty well. That's it's pretty much a standard practice, and it's supposed to be a stress reducer. How well it helps, don't know for sure, but it doesn't seem to hurt, but we don't think anyway. But kind of a standard practice.
Yeah, I think it even helps at that cellular level somehow with like changing the osmosis somehow. But again, I'm not. Yeah, that's about as far as I could go with that. I know it is pretty common. Katie, did you have any other follow-ups? I mean, you did some fish rearing, and maybe you all didn't do that in South Carolina, or?
Well, we were mostly stocking saltwater species, but when we did stock freshwater, like I'm familiar with like a saltwater dip for freshwater species of like, 5 to 15 minutes. But I just wasn't familiar with like that long of a transportation at 5 parts per thousand, which if that's standard practice, like that, totally makes sense. I was just kind of wondering what the acclimation was like to get up to that, like the acclimation period to both get up to that and then at the site of release to get back down to whatever the lake is at.
Randy,
I would
here,
guess there is, no acclimation. It's just a slight elevation just to help at that level. So it's far from the 35 parts per million, like in seawater. But yeah, I don't think there's an acclimation to that 0.5%.
Okay.
Nope, there's not.
Randy here, can you hear me?
Yeah.
Yep.
Hey,
Go
Randy.
ahead, Randy.
Oh, I was struck by the word that was used, that they're delicate, and I wonder if it'd be worthwhile to consider an alternative genotype as part of the experiment, specifically the Lake Huron intergress deepwater cisco, which I can show is very different than bloater in its ability to colonize Lake Erie, whereas the actual bloater that preceded it was not successful. It's just an idea. We can't really work it out here, but I wanted to park it more or less.
Yeah, that's a good point. This experiment is spurred off of the fish that are going into Lake Ontario and what we have access to, but that's a good question to bring up.
Yeah, that's good. Good question, Randy. And I left this webinar feeling like they're maybe not as delicate as we thought, given the mortality, the lack of mortality you saw in those, experiments 2 and 3. What do you feel like, Alex?
Yeah, it's hard to say. I mean, I certainly, after the net pen experiments and then seeing all the telemetry results from Natalie and Lydia and others, was expecting to see a lot of dead fish come out of the transport truck, and we didn't. But we kind of took them from their home and then put them back in their home. It's not the same thing as when you put them into Lake Ontario. They came back to a nice tank that was cleaned, and the water chemistry was exactly the same as when they left. And I'm sure that's probably adding to some of that mortality when we put them in the lake. So, like I concluded with, I think if you wanted to do this fully, you would have to put them into some type of net pens in Lake Ontario, or some type of holding container that had the same lake water with the same stressors, maybe mock predator, whatever it is. And I think it's just compounding stress. It's not necessarily just transport. It's like anything else when we study impediments. There's 1,000 of them, but which one can you control?
Yeah. Okay. Let's see. Go back to the chat there. Elizabeth Bradshaw is looking for details on the barotrauma mortality.
Yeah, that's one I haven't been involved with. Owen Gorman, I don't know if he's on the call or not. He's been working on this quite a bit, so he could speak to that, or Tim Johnson if he's on. I'm not sure, or anyone else that's been involved with that research. But the idea came from watching fish after they were released from the stocking vessels on sonar. They seemed to stop at a band of depth, and I can't remember what that depth was. But we're talking about fish that have lived their entire life in about a meter of water with no changes in pressure. So, potentially their swim bladders do not know how to react to intense changes in pressure like they would experience at a 50 or 100 meters. And so that was the idea behind that. But I haven't been involved with that research to know any of the specifics.
And do you remember, kind of building on that, do you remember, Alex, that Morton study that you summarized, or the Morton data that you summarized that was complementary to the net pen? Was there a high enough array around there to see if those, or was there ability to know if those fish, once they were released from the net pen, really went down deep to potentially have had that bara compression stress?
Yeah, I don't believe the acoustic tags they used had pressure sensors, so I'm not sure where the fish were in the water column. But the grid was huge. I mean, it was, I want to say 10 kilometers by 10 kilometers. And those fish were, I could go back to the figure, but they were detected over about 2 months. and there was almost no mortality until the end of the 2 months from that initial like shocking of, you know, losing 60% of the fish or 40% of the fish. Let me just bring it up. So, I mean, that was some support that the environmental conditioning had some effect on the fish. I mean, so mortality stops after the day that they were. Can you see that? Is that coming up on your screen?
Yeah, we see it.
So mortality stops here at like day 2, and then for 2 months there's no mortality in any of those fish, and then we start seeing predation. So this drop here is from predation, but also as fish start to leave the array, then he starts doing some different things with the probability.
Wow. Okay. Yeah. Thanks for showing us that again. I think I missed that the first time. Yeah. Cool. Okay. Yeah. Scott's got a good question asking you to basically build on what you said in your sort of discussion there, your concluding slide, thinking about eliminating transport stress altogether in terms of doing more in-lake rearing. Scott's asking you to kind of similar question. Have there been conversations to fertilize eggs, incubate them at depth in Lake Ontario?
Yep. Thanks for asking that question, Scott. I think you know the answer to it too, so I'll fill in for everyone else. A couple of years ago, we had the Fish and Wildlife Service and Ontario Ministry had a large amount of eggs that were not really assigned to anything. They said, "Do you want to do anything with these?" So we stocked about 1 million eyed eggs into Lake Ontario on different substrate types at those same depths that we had the net pen, so about 40 meters. And then we went back out with egg traps, or excuse me, larval traps to collect larvae as they hatch from the substrates. And we only caught 2 larvae in total. So out of 1 million eggs, we caught 2 larvae, and we learned a lot of what we did wrong in that experiment. And if we were to follow up on that, we would have to, you know, kind of really dig in on what pressure does to eggs, how to deposit eggs from 40 meters at the surface. We have not used specific like incubators that hold eggs and then you kind of release them, but that could be another method. Yeah, we're thinking about it and we've dipped our toes into it, but really haven't done a deep dive into how that would work. Thinking about in-lake rearing, we're mostly thinking about like, hard to explain, but like the large net pens that they have in the ocean, you know, for like salmon farming and whatnot. Basically finding areas in Lake Ontario where you could raise larval fish to age 1 and then release the fish from those large net pens. Is kind of the idea moving forward. But everyone knows it works in the Great Lakes. Spring is violent, and having net pens out in the lake, there's a lot of problems. Net pens collapse, they break, they sink, fish die in them. So we have to start thinking about those questions. That answer your question, Scott?
Yes, I wasn't trying to put you on the spot with that one, Alex. Sorry.
Did you know the answer to that? Did you know the answer, Scott?
No, I should probably have. Alex probably said something. I probably didn't have it at top of mind right now.
Okay.
No, I was thinking more about, you know, clearly it seems like barotrauma is playing a role at this fingerling life stage. And, you know, like you can correct me if I'm wrong, but aren't bloater fairly pelagic in their spawning behavior? And so that suggests that embryos are able to, decline in depth and survive that. I don't know at what speed. So I'm thinking, I don't know, maybe you could sort of avoid that barotrauma factor by, coming up with some sort of assay to release embryos to the bottom. I'd be happy to talk about some sort of assay to do that in an experimental setting. That'd be pretty interesting to chat about, but no, I wasn't trying to lead you any sort of way with that question.
No, I know you weren't. I was just joking with you. We did take eggs down in an incubator, drop them to the bottom of the lake for, I don't remember what it was, maybe an hour, bring them back to the surface, and then incubated them at the Tunison lab to see, like, did that blow up eggs? What happened? And all of those eggs hatched perfectly fine. So barotrauma, at least we didn't look at every physiological effect, but at least the eggs had hatched that were dropped to that depth and then brought back. So that was one piece of information. I think the eggs would be fine. It's just getting them to survive from, whatever day you put them down until the day they hatch.
Right.
Yeah. Thanks, Scott. Alex, I mean, you were mostly like stocking, or your experiments were mostly yearlings, right? And I know there's been a history in Lake Ontario of stocking some fingerlings. I think that's right. Some fingerlings and then some yearlings, and they've sort of settled on yearlings. I don't know if is that fair? If Steve is still on.
Yeah, I think the early days of stocking was fall fingerlings, and then kind of following the lake trout approach, they went to age one.
Yeah, whenever we were doing the Cisco work, we were doing fall fingerlings, and then ever since we've been doing bloater, I think it's been a spring stocking. So basically 1-year-old fish, or 13 months, 12 to 13-month-old fish.
Yeah, I'm just thinking about like what we've learned with Cisco, our Teddy, and Saginaw Bay reintroduction. It looks like the spring, they're like May 45, 50 millimeters seem to survive better, or at least come back. But let's just say they survive better. So getting them in the lake earlier. I mean, I've heard hatchery guys talk about sort of how the younger fish can handle stress better. But for bloater, that's harder because they're hatching so much later than our Teddy. So it would take them until fall really to get that size, and there's not as much food to eat in the fall as maybe there is sort of as the spring zooplankton is building up. But I do like the idea of trying to get them in the lake as soon as possible. But I don't know if there's a sweet spot there where they can get large enough to be able to be transported and still enter the lake at a time when it's somewhat stable and has a lot of food.
Yeah, I think Brian Whitehall had talked about playing around with water temperatures to try to get them to grow super fast, which we were also at the same time trying to figure out how to grow fish slower and more natural. So it was like, well, which one do we want to do here? Grow a big fish really fast or grow them like a natural progression?
Yeah, exactly. And that's what they're having to do with those spring Cisco is they grow them, they
Right.
incubate them way warmer than what they typically would experience. Yeah. Yeah, Katie.
I was just wondering, with concerns about barotrauma, if anyone had looked at swim bladder morphologies and comparing hatchery reared and wild bloater at different ages, and maybe even like between fall stocked fish versus spring stocked fish, just to look at like what the actual physical capabilities of the swim bladder are by looking at wall thickness and things like that.
Yeah, I think that's part of the research I was talking about earlier that Owen was leading. Tim Johnson had give us some little tidbits about he was basically talking about heart attacks being caused by barotrauma, and that's maybe what they were seeing as like the mechanism that kills the fish. And I think they were looking at swim bloaters as well, but again, I don't want to talk out of turn here because I wasn't involved with it, but I think there are folks that were or are looking at that.
Okay, thanks. And sorry if I missed
Yeah.
that and you were talking about it earlier.
You
Nope,
didn't.
just no.
Yeah, I can't add much more to that either. I feel like Owen has talked about his work before, but if not, we might grab him for a future webinar. All right, any other questions out, Ben?
Hey, Alex, great talk. I've got a couple questions. One is the first one is so the cortisol stress related stuff you tested. What I know not all studies are exactly the same, but is bloater higher or lower relative to other species that are stocked, like lake trout, walleye? We've got a pretty good handle on stocking them. So I was wondering, one, what's the bloater cortisol levels relative to other commonly stocked fish? And then second one, so I know the cragnines that are raised in a hatchery setting, they can have that pug headedness. And I was wondering if maybe they all have it, but did you notice any relationship between ones that were dying having it, and then maybe some that were living not having it, or anything like that?
Yep. Thanks, Ben. Good to see you. Thanks for the questions. To answer your first one, as far as cortisol goes, it seems like a lot of these studies, these things are species specific as far as like what, not even to call it threshold, but like where they hit a point of like with an acute stressor. As far as baselines go, like 0 to 20 nanograms per milliliter is pretty average for a base of like a lot of cold water fish. And most of this research is done on, you can probably guess, rainbow trout and other Pacific salmonids. In some of the studies that I've read, they're like acute cortisol threshold is like 100 to 100 or 100 to 300 nanograms per milliliter, where bloater weren't even close to that. But the studies are set up differently, and it's a different species. So I'm not really sure how it compares to lake trout. As far as like a one-to-one comparison, I'm not sure that we can make that comparison. So hopefully that answers a little bit of your question. The second question about pug-headedness, we kept every fish that had died after it came back to Tunisia, looking for exactly that, like what is something that we could tell maybe killed the fish. There was nothing with pug-headedness. Fish that had abrasion were usually the ones that were pulled out of the tank dead, so they had, like loss of scales or something, or they were crushed in the tank in the truck. But beyond that, we didn't do any dissections to look at like physiological changes in the fish. So I'm not, yeah, we didn't see that relationship. I don't know. I think Hansi at one point was on this call. He might still be. He might have some more information about that, because I know he studies pug-headedness, but we didn't see that in this experiment.
All right, cool.
Like a Purdue alumni event here.
Reunion.
I just told Andrew he has to ask a question. Make this a lab meeting.
No, don't ask any questions, Hansi. Won't be able to answer them.
No questions from this quarter. Yeah, I'm not aware of. There are morphological differences that we've identified, as Ben alluded to, with bloater and with Cisco, with hatchery reared versus wild fish. And we're still, I think, in the process of investigating sort of what that does to survival fitness when stocked into the wild. So no definitive answers there yet.
Thanks, Andrew. So a few more things in the chat, not questions, more comments. Kevin Keeler talking about other higher concentrations of salt tip and their use for treatment, different than what we were talking about for transport. And then Bruce Morrison from Ontario, just giving a thumbs up in terms of how this work has been developed. And he was in early days of the bloater reintroduction way back to 2009. So a good attaboy from Bruce. All right. Well, that's a lot of questions. Great discussion. Thanks for hanging on so long, Alex, and to like almost 50 of you here over an hour and 10 minutes into the webinar. And great participation today. And we look forward to having you back next month. It's about, again, that'll be middle of the month. Renee has dropped a reminder there to register for that webinar. I think that'll be super interesting by Claire Venny. But if you're still on, please give Alex a virtual round of applause, and send any further chats of appreciation. Until then, thanks again, everybody, and great job, Alex. Appreciate it.
Yep. Thanks for the invite. Thanks everyone for hanging on so long. Appreciate it.